Good morning everyone. In this video, I'm going to tell about the anaphase. Ana means up, phases means appearance. Anaphase. Anaphase begins brutally with the synchronous splitting of the chromosomes into two chromatids which are called as daughter chromatids. Daughter chromatids with each one kinetochore. Synchronous splitting of the centromere during prophase is evidently caused by increase in the cytosolic calcium ions. In fact, calcium containing membrane vesicles accumulate at the spindle poles, release the calcium ions and initiate the anaphase. One set of anaphase, each chromosome arranged at metaphase plate, split simultaneously, and the two daughter chromatids, now it is called as chromosomes. Now it is referred to as chromosomes of future daughter nuclei, split simultaneously, begins migration towards the opposite poles respectively. Anaphase involves the following two steps. One is anaphase A, another one anaphase B. So these two will be. During it, there is a poleward movement. There is a poleward movement of the chromatids due to shortening of the kinetochore spindle. Kinetochore means which is attached to the kinetochore, that is called kinetochore spindles. During their poleward migration, so that chromosomes characteristically appears in different shaped J shape, I shape, U shape, V shape, like that. Different shapes will be. Metacentric means the arms will be either side will equally. So there is no difference between that. So equally they are attached with the centromere. So that is a metacentric. Submetacentric means what will be, how will be. So one side very short, one side little long. This is called submetacentric, means one arm is very short, one arm is very long. And acrocentric. Next, acrocentric means very short and centromere, with the centromere, and another one is very long. So like this will be. This is called acrocentric. So another one telocentric means very short, that is completely terminal, it is not visible at all. So only it look like a one arm attached to it, like that there will be. So the different shapes we can be seen here. Anaphase B, it involves the separation of the poles accompanied by elongation of the polar microtubules. Aster microtubules also held in anaphase B by their attractive interaction with the cell center. When we zoom it, so these chromatids are telomere begins to break themselves and pulling and pushing with the either respective poles. So generally interzonal poles are involved pulling and pushing either sides. This is the best phase to see the chromatids in different shapes. The process of anaphase will continue till the all chromatids reach their respective poles. At onset of anaphase, there are three proteins which are involved in the moving of this chromosome. These are kinesin 13, kinesin 4, and dynein. dying, dying. These proteins as well as kinesin 4 also inverse. So there is a these kinesins are one side, dynein in one side. These are pulling and pushing either sides, moving force to either side equally. Kinesin is a positive interactive protein and dynein is a negative interactive protein. First one, centromere splits and chromatids separated. Then these chromatids moves either sides of the poles. As each chromosome moves away from the equatorial plate, centromere of the each chromosome towards the poles and ends at the leading edge with the arms chromosome training behind. There are two events in this anaphase. First, as each chromosome moves away from the equatorial plate, centromere of the each chromosome towards the poles and ends at the leading edge. First one, centromere splits and chromatids separated. Then these chromatids moves either sides of the poles. As these chromosomes moving towards the pole, centromere will be foremost. Here also, southern pole, when they are migrating, centromere should be foremost. Later, these chromatids will be. These, when they are pulling either sides, they are bipolar forces moving, pulling and pushing, then the stretch will be equally. Otherwise, what will be? These chromatids, more chromatid one side, less chromatid this side means that is abnormal conditions occurs in that. When they are giving the motive force, kinesin 5 and kinesin 7 also involving in this movement of the chromatid movements either side, and ATP also energize the pulling the either sides. Then only it is possible to moving either sides of the poles. They are stretching either sides equally. Then only it is possible equal distribution of the chromosome. After reaching the poles of either sides, the decondensation of the chromosomes occur, that is begins to the telophase.